Hi class, welcome to the online session. We are now going to proceed with the third part on Neoplasia chapter. So we look into the growth promoting metabolic alterations that can occur among tumor cells. So before we look into it, let's look into the other metabolic activities that other cells would do. So in quiescent cells, usually they would undergo oxidative phosphorylation for the generation of energy. In this case, there would be the formation of 36 moles of ATP with less formation of metabolites because it is not needed during quiescence. But in normal growing cells or tumor cells, they would require aerobic glycolysis, which is Warburg effect, to be able to get the more important metabolic intermediates that would be used for formation of cellular constituents like proteins, like lipids, like nucleotide, those for nucleotide synthesis. And in aerobic glycolysis, it will only form 2 moles of ATP. Energy in this case is not the priority. The priority here is the formation of metabolic intermediates. So, with regards to normal or normal growing cells or tumor cells, they would rely on the same form of metabolic activity, which is aerobic glycolysis or Warburg effect. Then what would be the difference between tumor cells and that in normal growing cell activity? And this is the presence of metabolic reprogramming that is seen in tumor cells. When you say metabolic reprogramming, this would refer to the mutation on oncogenes and tumor suppressor genes. So in this case, if we have mutation towards the oncogenes, it will lead to cellular proliferation. Okay. And if there's the suppression of tumor suppressor genes, and this would lead to transcription that would lead to cellular proliferation. So the signaling factors that would be used here would be the PI3K, AKT signaling pathway. We have the we have here the tyrosine kinase receptor, and then we have the overexpression of the MYC. So the PI3-AKT signaling pathway is used to increase the transport of glucose into the cell, and it will lead to increase in glycolysis, and then there would be the lipogenesis and protein synthesis. Uh, in the case of the reverse of the receptor tyrosine kinase, uh, it tends to attenuate the activity of the pyruvate kinase, M2 isoform within the cell, within the tumor cell per se. And this will cause the buildup of glycolytic intermediates that would then be used, okay, for the formation of amino acids and proteins, the DNA and RNA that would lead to growth. Overexpression of MYC in this case would lead or would promote anabolic metabolism and cell growth. So, by the way, if tumor suppressor genes are not mutated in tumor cells, or in the case of normal growing cells, the metabolic activity here would be reduced, okay, or would uh, make the activity inactive by decreasing the signaling of PI3-AKT pathway by PTEN, STK11, and P53, which by now you know that they are tumor suppressor genes. So next we go to the evasion of apoptosis. Okay, so in evasion of apoptosis, this is what tumor cells would want to have in order to achieve unrestricted growth. So there are two pathways for apoptosis to occur. So One, we have the extrinsic pathway here, which is which would then be activated by the binding of the CD95, which is the FAS receptor or the death receptor to the FAS ligand. Okay? And this would lead to the trimerization of the receptor attracting the adaptor protein FADD, which then would recruit the procaspase 8, and it will generate the formation of the death-induced signaling complex. And this will activate caspase 8, and it will activate caspase 3, and it will lead to apoptosis. On the other hand, if we have the intrinsic pathway, which is related to the formation of damage to the DNA caused by stress, radiation, or chemicals, this will elicit a response from the P53. Okay, so once DNA damage has been sensed, it will then lead to the activation of pro-apoptotic genes, which would be the BAX, BAK. Okay, uh, and it will uh, inhibit the actions of the anti-apoptotic genes like BCL2, BCLXL, and MCL1, leading to mitochondrial permeability with the secretion of cytochrome C and APAF1, which will promote the activation of caspase 9, activation of caspase 3, and finally apoptosis. So tumor cells would now try to evade apoptosis through the following mechanism. Number one is to limit the P53 response through suppression of of this tumor suppressor gene. So if there would be the inactivation, there would be no sensing, and there wouldn't be any apoptosis. Number two is try to look into the mitochondrial factors. So what are they? So we have here the pro-apoptotic gene, and this would be the anti-apoptotic genes, the BCL2, BCLXL, and MCL1. So what happens here is tumors would favor, of course, the anti-apoptotic genes. Okay, so they would overexpress these genes. And then next we have the caspase 9, which there would be the inhibitor of caspase 9, which is the inhibitor of apoptosis protein or IAP. And then we also have here the formation of a flip. Okay, that binds with a death-induced signaling complex and prevents the formation of caspase 8. So those are the different steps or methods to which tumor cells would be able to evade apoptosis. So next we have here, this is an illustration that shows to you the balance that would be needed in normal conditions, okay, in order to prevent untoward cell death or cell survival. So here we have, what's this one? These are the anti-apoptotic genes, okay, that would promote cell survival, like BCL2. On the other hand, here we have the BAX, okay, BAK. And these are the ones that would prevent, they are the ones that would promote apoptosis. So these are pro-apoptotic genes. So next we go to limitless replicative potential. So this would refer to the activity of telomerase, which is present in stem cells but would be inactive in somatic cells, or would be low in activity. So for example, this would be the somatic cells. They would possess inactive telomerase. So they are only given up to 70 doublings or cell divisions before they would present with shortening of the telomeres. So telomeres would be the end portion. So in this case, if there would be the presence of shortened telomeres, this would be sensed by the p53 if it is unmutated, leading to the senescence of these cells. However, with mutation of the p53, it will lead to the activation of the non-homologous end joining pathway, leading to the formation of dicentric chromosomes. It will lead to it will go into mitosis and will lead to the formation of new double-stranded breaks. This will continue on forming a bridge fusion breakage cycle, which if it will continue on, it will lead to mitotic catastrophe or mitotic crisis. Okay, so this would occur, the mitotic catastrophe would occur in cells that will have inactive telomerase activity. But because of genomic instability, the telomerase activity can be reactivated or can be activated in somatic cells and can lead to malignancy. Next, we have angiogenesis, which is another very important factor for tumor growth. So tumors would require, when they are going to grow to more than 1 to 2 millimeters, okay, they would require the presence of a supply, oxygen, for nutrients, as well as the removal of their wastes. Okay, so the formation of blood vessels would be triggered by the presence of proteases in the area, as well as the presence of hypoxia. So the moment that we have these two, okay, especially the hypoxia, it will lead to the activation of the HIF1 or the hypoxia inducible factor 1 alpha, which is an oxygen sensitive transcription factor, and it will activate the vascular endothelial growth factor as well as the basic fibroblast growth factor, leading to the formation of new blood vessels, okay, and it will promote growth of the tumor. Okay, so how does our body react to this one? So there are tumor suppressor genes, okay, that would possess ability uh, to stop angiogenesis, like p53. It possesses anti-angiogenic molecule like thrombospondin 1 that would prevent VEGF overexpression. Okay, it would prevent VEGF overexpression. Okay, so next we go to the um, invasion of the extracellular matrix. So the invasion of the extracellular matrix would start with the loosening of the intercellular junctions, like this one, like the id-cadherin. There's downregulation of the id-cadherin, like in adenocarcinomas, uh, and this would be in preparation for the next step, okay, which is degradation of the basement membrane. In this case, there would be secretion of proteases, okay, like type 4 that can cleave the stroma and degrade the collagen. And then this would be followed by attachment. So tumor cells, okay, which are already loosened from the addition to the other epithelial cells, would then attach to the receptors found in the stroma, like the fibronectin and the laminin. Okay, uh, and this would be used for the next step. Okay, which is migration. So this is the final step, wherein this step is mediated by the cytokines such as the autocrine or autocrine motility factor, uh, with subsequent detachment from the matrix by cleavage products, resulting to chemotaxis. Okay, chemotaxis. Okay, so this is the hematogenous spreading model wherein once the tumor cells have intravasated or has gone within the blood vessel, they would tend to form aggregates or clumps. So this aggregation or this form of adhesion can be among tumor cells, which we would call as homotypic adhesion, okay? Or they can be an adhesion with platelets like this one, with lymphocytes, or with coagulation factors. In this case, they tend to become tumor emboli. The extravasation like this one into the distant sites would require the possession of adhesion molecules like the CD44 that would be derived from the T lymphocytes would then favor lymphoid tissues and other sites. So organ tropism would be related to ligand preference. Okay, when you say organ tropism, it refers to where the tumor emboli would go. Okay, so it would be related to the ligand preference to endothelial cells that would line the area of the distant sites. It can be associated also with chemokine expression in tumor cells. And lastly, there are some sites that tend to be unfavorable, like the skeleton, a skeletal muscle, not the skeleton, na skeletal muscle in the spleen, uh, and they rarely act as metastatic sites. So some of these tumors may opt for small, insignificant metastatic sizes, and they would stay there for a couple of months or years, and they would adhere to the concept of dormancy or prolonged presence of micrometastases into a distant site without progression. Okay. So next we go to the molecular genetics of metastasis development or metastatic variants. So here are the different mechanisms that would explain the occurrence of metastasis uh, among tumors. So number one, so we have the letter A here, referring to the formation of metastatic variants. So when you talk about metastatic variants, this would refer to the formation of those variants from a primary tumor due to the formation or accumulation of several mutations, which tends to make it heterogeneous. And this would be the clonal evolution model. For letter B, it would say that there would be the formation of the gene expression, which is called the metastatic signature, which has been there, okay, ever since. So it means that they already possess the genes for metastasis. Okay, so no subcloning required. And letter C would be a combination of A and B, which would mean that we already have a metastatic signature present, and there would be subclone formation, okay, leading to metastasis. And another model would be the stromal response. So the stromal response would be a great help or would be a prognosticating factor for the regulation of the growth, invasion, and resistance to metastasis. So with regards to metastasis, there are genes that would be geared for metastasis. These are the metastasis oncogenes, snail and twist, to which they would promote EMT or epithelial to mesenchymal transition with loss of E cadherin. So now let's go into tumor immunity and how does the immune system detect the different tumors. So tumor antigens are formed by tumor cells, and they would cause the immune response. So these are the different antigens that would be expressed by tumor cells. So one would be the product of mutated genes like oncogene or mutated or tumor suppressor genes. Another is the presence of mutated self-protein. Another would be the presence of overexpressed or aberrantly expressed self-protein like the tyrosinase, GP100, MART1, and we have the MAGE. And then we have products of the oncogenic virus like E6, E7 that we see with human papilloma virus. And then there are antigens that uh, would not be targets of anti-tumor immunity. So it means that they simply would be there in high levels, but they do not cause an immune response. Example of this oncofetal antigens would be the CEA and AFP. And then we have the altered cell surface glycolipids or glycoproteins, which tend to be higher in value in cancer than in normal cells. Examples of which would be the gangliosides and mucin. And lastly, we have the cell type specific differentiation antigens, which can be seen or would be called as clusters of differentiation. If you remember in histology, as we have the CD for the different lymphocytes or leukocytes, and these are specific for a lineage of for identification of monoclonal expansion. Like in the case of a B cell lymphoma, that is a CD20, and if it's a T cell lymphoma, that's a CD3. Okay. Uh, Next, we go to how tumor cells or tumors would evade the defense. So we have anti-tumor effector mechanisms wherein they would form evasive maneuvers. So what are these evasive maneuvers? By the way, uh, the immune response would be taken care of by by cells such as the cytotoxic T lymphocytes, okay, like the natural killer cells, like the macrophages and monoclonal and then we have the monoclonal antibodies that would be against the antigens. Okay, so what are the different evasive maneuvers by tumors? So first is the formation of an antigen negative variant, okay, or an antigen that would mask uh, uh, that would be masked by glycocalyx molecules, okay, and it will cause lack of T cell recognition. Another is the formation of a reduced expression of theirs, or there is loss of MHC molecule expression. So in the same way, it will lead to a lack of T cell recognition. And then we also have the formation of immunosuppressive proteins like the TGF beta, which will lead to T cell inactivation. And then we have the activation of inhibitory receptors uh, for T cell activation, like CTLA4 and PD1. So again, if you have CTLA4 and PD1 activation, it will lead to inactivation of T cells. Okay, and lastly, we have the presence of Tregs, which refers to immunosuppressive regulatory T cell, which would aid in immunoevasion. So next, we go to genomic instability with regards to DNA repair defects. So there are three types of DNA repair proteins. We have the mismatch repair genes, we have the nucleotide excision repair gene, and we have the recombination repair genes. Individuals who are born with DNA repair defects in any one of them would be at risk for development of cancer because this will allow genes to mutate as they are not repaired by defect by mutated repair genes. So let's start with hereditary non-polyposis colon cancer syndrome, or the HNPCC, or they would also call it as the Lynch syndrome. And this is an autosomal dominant disorder with increased frequency of colorectal cancer and is associated with the development of ovarian or endometrial cancer. So this is a defect of DNA mismatch repair genes, and it follows the Amsterdam criteria when we look into HNPCC patients. So we look into their lineage, wherein there should be the presence of three or more relatives who had colorectal cancer, and one of whom is a first degree relative of the other two. Another is we look into the age. So the colorectal cancer cases within the family would be diagnosed before the age of 50. And lastly, colorectal cancer would involve at least two generations. So which repair gene would be involved here? So the repair gene MLH1 and MSH2, which when they have mutations, they would cause microsatellite instability because they are used in proofreading, and it will show tandem repeats of nucleotides, okay, in creating varying length, creating new alleles. Next, we have the seroderma pigmentosum. So this is an autosomal recessive disorder associated with defect or defect in DNA repair for nucleotide excision repair gene. And the problem here is that the nucleotide excision repair is responsible for the repair of UV damage during the course of exposure to UV light. Like if we're going to do a sunbathing, or uh, we go from one place to another, exposed to the sun, the UV light upon exposure to the skin will lead to cross-linking of pyrimidine residues. And this would be repaired through the NER or nucleotide excision repair. Okay, with seroderma pigmentosum, they should not be exposed to UV light, okay, or else it will lead to cancer. Okay. And then the last type of DNA repair would be by homologous recombination. So in this case, this would be a group of autosomal recessive disorders that would be characterized by hypersensitivity reaction to DNA damaging agents like ionizing radiation or to chemotherapeutic drugs. So like for Bloom syndrome, they are sensitive to ionizing radiation. It will lead to developmental defects. For ataxia telangiectasia syndrome, you have the neural syndrome, and then we have the Fanconi syndrome, which has sensitivity to chemotherapeutic drugs, leading to bone marrow aplasia. BRCA1 and 2, if you remember, these are tumor suppressor genes. They are involved in familial breast CA and involved in estrogen receptor regulation. Why is it present here? Because it makes a network with Fanconi anemia proteins that will promote homologous recombination DNA repair. However, if there would be mutation towards the network between BRCA1 and 2 as well as the Fanconi anemia proteins, it will lead to the opening of the non-homologous end joining pathway, and it will lead to massive aneuploidy. And then we have lymphoid markers or lymphoid genes like RAG1 and RAG2, as well as the AID or AG induced cytosine deaminase, uh, which these are genes that would act as homologous recombination genes. And if there would be mutations towards these genes, it will lead to lymphoid neoplasms like lymphoma. So that's my last slide for this lecture. Kindly read your Robbins for more information and details. Thank you and good day.